All right, so this video will focus on how DNA replicates. So replication is just how it makes another copy of itself. The first question we should ask ourselves, though, is why is DNA replication important? Like, what is it used for? And so if you look at this image, you actually see a cell that's dividing into two. And we talked about this in terms of the cell cycle. In order for growth and maintenance, you need cells to divide and to form more cells. And so in this cell, you can't see the nucleus, but there's going to be DNA, right? One copy of DNA. But when we make what we call daughter cells, you need to make a second copy of DNA. So you have one copy here, and then one copy here. In order to get those two copies, before you divide, you need to duplicate that one copy or replicate that one copy, so you get two copies. And so if you remember the cell cycle, we have G1, this is interphase, right? G1, S, and G2, right? And if you remember during the S phase, this is where DNA replication occurs. Before you can divide, you need two copies so that each daughter cell gets the copy of their own. A cell without DNA is not going to be any good because there's no instruction manual to make proteins, and then so that cell will eventually die. Okay, so that's why we need to replicate DNA. The question now is, how does it happen? So here's some basic information. Replication is when DNA copies itself exactly. During that process, mistakes are actually made. And so when mistakes are made when you replicate, that means it's not an exact copy, and so we call those mistakes mutations. All right. When they replicate, they know that they have to have what we call complementary base pairs. So like A is always T, C with G, and so let's go through the steps. So the first thing is it's got to uncoil and unzip, and so it's normally as a double helix, as you can see here, and so there's an enzyme called helicase that actually unzips it, so it exposes the base pairs and actually pulls the base pairs apart. So now they're exposed. Then what happens is, as you can see down here, what happens is they start, there's something called DNA polymerase, another enzyme that starts adding new bases and a new strand. So what you can see is it starts, since now these base pairs are separated, a new strand, and you can see right here, a new strand is put on. And so what happens is new bases that correspond are attached. And so the other strand, this is occurring as well. And so now you're making two new strands, and then you can see it coils back up again. It twists back up into a double helix. So from this, you started with one strand, and then now you have two strands. Well, I shouldn't label it like that. Let me do it like this. So you have total of two strands, right? So here's one strand, and then here's your second strand. Okay? All right. So our second step, so our first step in DNA is separate using helicase. The second step is to make a new strand of DNA, and that binds with the old strand. Okay? And so now you have two copies of the DNA. And then the last step is you have to proofread, right? So you got to separate everything. You actually got to do the work at step 2, and then you got to proofread to make sure it's accurate, right? So there is DNA polymerase also double checks what's going on, but errors still happen. I mean, you imagine like you have 3 billion tasks or bases you have to match up. 1 in a billion, there's going to be a mutation. A lot of times it corrects, but sometimes it just misses it, right? Just imagine like you There's always going to be a mistake somewhere. When you proofread something, you just miss it, right? And so it happens. It happens here. Okay, so the last term we got to learn is that DNA is what we call semi-conservative. And so what you'll notice is that if you look at the two new strands, or the two new DNA copies have one old and one new, right? And so we call it semi-conservative, meaning like you conserve one and then you have a new DNA strand. So you have the old strand, which is called the template, and the new strand is called the complementary strand. Okay? So we call it semi-conservative. All right.